Howdy.
Howdy.
Yes, my name is Reghan Mutethia. I'm a PhD candidate in Bashir's lab here in Texas A&M. And today I'm going to present about the transcriptome study, which we have done and published this year in August on 60 gene reprogramming rosetted and symptomless shoot from the same mature rose plant infected with rose rosette virus. So as we know, the rose rosette virus is so interesting because we know it causes rosetting on one branch, whereas the other branch looks healthy. And then, so these are when you have a close look of the rosetted branch, you see some the symptoms here: the hypodoniness, deformed leaf, deformed flowers, and decaying stem. So these are the plants we used for our study where we did this transcriptome. So we collected samples from the rosetted leaves of the branch, and then we collected the samples from the asymptomatic, and then we collected the samples from healthy. And all these plants, they were grown, they were in the field here in Texas. So as we know, like this is how because it was infected by the field infected plant, so it was transmitted through the area of mite. So after we collected the samples, we did the normal PCR, and as you can see, the real-time PCR data shows like the symptomatic or the rosetted tissues, they end up decombined compared to asymptomatic, and the healthy plant didn't have any bad. And on this real-time PCR, we focused on primers for RNA3. As you can see on the active control, we use the same concentration for these samples, and they were more on virus levels on symptomatic compared to asymptomatic. So we went ahead to look at the virus status for these samples, and surprisingly, all in the virus levels using the qPCR, there were more high virus data in symptomatic when we use primers from RNA3, RNA5, RNA6, and RNA7. So the virus data was more on symptomatic compared to asymptomatic. And if you look keenly, you can see the virus difference between symptomatic and asymptomatic in RNA3 was more than 550-fold change in RNA3. When you look at RNA5, they were more than 115-fold change. RNA6, they were fewer at least 60-fold change, and RNA7, they were like 2-fold change. And from this data, we can take the takeaway we can have from this qPCR and real-time PCR is like the primer3 is more sensitive for detecting RNA3 for detecting RLV. As you can see, there is more fold change. The other thing we can take from this is RNA6 and 7 is more it is critical, as it can be critical as well for the infection, because you can see as well there are some fold change difference between symptomatic and asymptomatic. And then this data as well suggests that the higher virus levels in symptomatic shoot may be influencing the rosetting. And the RLV is, and we can hypothesize because the RLV is more on symptomatic compared to asymptomatic, we can suggest that there might be a blockage which is preventing the virus from moving from symptomatic to asymptomatic. But even though the asymptomatic branch is showing healthy, it is infected as well. So we went ahead and did the transcriptome analysis with these samples, and after doing the transcriptome, we got the data, and after analyzing the data, we compared the differentially expressed genes between the asymptomatic with the healthy, and you can see the levels of the differentially expressed genes in asymptomatic are almost equal between asymptomatic and between downregulated genes and upregulated genes. And then we went ahead and then we compared the differentially expressed gene in symptomatic compared to healthy, and you can see the number of genes are almost equal in which are downregulated and upregulated. we went ahead and compared the genes, differentially expressed genes in symptomatic to asymptomatic. And we can see here the number of genes which are upregulated in symptomatic were more compared to asymptomatic. So we went ahead, as you can see in panel B, to try to identify these genes which are represented in this volcano plot. And we got a total of 1509 genes which are upregulated in asymptomatic disk conditions. And if you go into deep into these genes, there were 739 genes which were upregulated and 770 genes which were downregulated, which are represented in this volcano plot. And then we went ahead and trying to find out which are these genes which are represented in this volcano plot. And we come to we identified 1484 genes which were differentially expressed in symptomatic. And when we went ahead to look at the downregulated genes and upregulated genes in this 1484, we came to get 874 genes were upregulated in symptomatic, and then 610 were downregulated in symptomatic. So we went ahead to because we have these genes, so we want to see the genes which are either shared between the conditions or the genes which are distinct. And it was so good to see we have some distinct genes which are not shared by any conditions. Like we can see we have 232 genes which are only upregulated in asymptomatic, and we have 332 genes which are only upregulated in symptomatic. And finally, we had a bunch of number of genes which are shared between asymptomatic up and asymptomatic down, and those total number of genes were 545. And then when we went to look at the downregulated genes, we had 344 genes which were distinct only on asymptomatic, and we had 219 genes which were only distinct in symptomatic. And we had 389 genes which were shared between asymptomatic down and symptomatic down. And then we looked at the genes which were oppositely regulated, which were more interesting when we saw them. And we found there were 37 genes which were symptomatic up, and they were shared with asymptomatic down. And this, when we saw this, before we started to look into details of what the function of these genes, we were so happy because it would answer the cause of rosetting which we see in symptomatic. And then we got two more genes which were asymptomatic down and symptomatic up. And so when we went to look at the details of these 7 and 2 genes, they were now focused more on the developmental side. So that's why we went ahead to look at the function of look into analysis of to analyze the function, all the genes in this group. So from there, we went to look at the gene ontology so that we can categorize according to the biological roles which are involved in development for all these genes in our data set. And then we contrasted the geo terms, which are in either asymptomatic and symptomatic. And what we expected, we were expecting to have a unique terms in the certain or symptomatic shoot, but we started to look at the gene terms or geo terms, which with higher gene ratio and FDR values in both, and we found there were 3 genes which were shared in either asymptomatic and symptomatic, and these were shoot axis formation, the secondary shoot formation, and morphogenesis of branching. And then we went ahead, because this didn't tell us like what is the source of rosetting, we went ahead to look at the asymptomatic related terms. And the terms which we found were endodermal cell differentiation and negative regulation of nuclear division. So these were only downregulated in asymptomatic. So we went ahead to look at the terms which were uniquely upregulated in certain tissues where we see the rosetting. And if you can look keenly, the terms which were upregulated in certain tissues, they were seed development, fruit development, and tissue development, reproductive structure development, system development, and formation of plant organ boundary. And if you look in at these terms, they are involved in development and they appear on the upper part of the plant. So the takeaway from this gene ontology on the developmental terms, biological terms, is the setting include factors functioning as the shoot apex and the reproductive development of the plant. And the setting might be caused by manipulation of genetic factor by R&B, as we can see here. So we went ahead to classify the differentially expressed gene represented in these biological terms, and we found that chromosome 2 genes are the one which are associated with women are highly downregulated in infected samples. As you can see here, these are the total number of downregulated genes which are associated in chromosome 2 in both asymptomatic and symptomatic down. And then and then when you have a close look, we can see we have genes that are associated with chromosome 5, 6, and 7, which are downregulated in asymptomatic compared to symptomatic. As you know, like these genes associated in chromosome 5, 6, as Oscar Lab group work on the QTL, they have identified that these genes are involved in defense. So, because this paper is more focused on development of rosetting, so we were more focused on the genes which are associated with development, which is involved in chromosome 2. And as we know, like the genes which are involved in chromosome 2, they used to control and development, and there are a couple of research which shows like chromosome 2 is involved in research, as you can see here. And yes, and then chromosome 5 and 6, I have said it is involved in defense. So we went ahead to look at the developmental hormones, how they are manipulated by RLV leading to programming of plant growth. As you know, like the plant hormones are produced at specific regions within the plant, and then they are transported to other parts of the plant for action and storage. And the hormonal crosstalk between the pathways and the complexity in understanding their roles. So we expected the developmental hormones would be altered in rosetted. And for the developmental hormones we looked at the auxin, gibberellin, streptoractin, cytokinin, and abscisic acid because they involve in development. So we expected them to be altered by RLV. And the characteristic of RLV, as we know, is abnormal formation of leaves, abnormal clustering of the branches, altered flower morphology, thickened branches, and hyperthoniness. And as you can see here, they appear on the upper part of the plant. So we are expecting now the developmental hormone to be altered on rosetted shoot of branch. So we started to look at the gibberellin and caffeic acid, and we found they are manipulated in infected plants. So when we looked at the auxin levels or IAA, we didn't see any difference when you measure the hormone levels. So they were equally produced in the plant in healthy, asymptomatic, and symptomatic. So when we looked at the gibberellin levels, we found gibberellin or GA, they are more produced in asymptomatic compared to symptomatic at healthy. And when we looked at the levels of ABA or abscisic acid, it was slowly produced and equally in healthy, asymptomatic, and symptomatic. And then we looked at the caffeic acid levels. It was more produced on symptomatic compared to asymptomatic and healthy. So as you know, like the caffeic acid levels, which is more on the symptomatic compared to asymptomatic. So caffeic acid is involved in lignin peroxidases, which leads to stem thickening. So and then the takeaway from this slide is like the virus data illustrated may be altering GA and caffeic acid. As you can see here, the levels of caffeic acid is more symptomatic and the level of AVA is low on symptomatic. So we went ahead and looked at the gene ontology of developmental hormones, which are associated with development in asymptomatic and rosetted tissues, and the auxin levels the auxin levels are similar between the differential expressed genes and the GCMS. So what we mean here is when we measure the levels of auxin, it was the same in asymptomatic and symptomatic, and the DEGs, as you can see here, is the same. So the upregulated geotherms in certain tissues, they were response to gibberelline, which we have seen it is altered by the LLP, and then the gibberelline mediated signaling and response to endogenous stimulus. And surprisingly, as you can see, we had Stigoractone, which is almost like a new hormone, which was upregulated, which was upregulated the geotherms in both asymptomatic and symptomatic, and their gene ratios is higher compared to other geotherms. And as we know, the Stigoractone represents the outgrowth of auxiliary bands, and we think this might be inhibiting the function of the auxin. And all these hormones which we have worked on them here, they involved in leaf development, flower development, and plant bud development. So we went ahead to manually analyze of individual hormone pathways so that and it reviewed the auxin and caffeic acid that specifically manipulated in prostated and asymptomatic tissues. So we worked on we identify 18 genes which are associated with the auxin and caffeic acid metabolism. As you can see, these are the 18 genes which we worked on. And first we started to look at the caffeic acid. And in the caffeic acid, we found one gene which is caffeic acid methyltransferase, that is commit gene, which is involved in production of the caffeic acid. And this gene is more upregulated on symptomatic, where we have thickened stem. And when you look at the heat map, the caffeic acid is more the levels of application is so high that it's almost like 8.0, the highest one. And then in asymptomatic is no. So when we came to look at when we measured the level of the caffeic acid in the qPCR, we found the levels of caffeic acid is more on symptomatic compared to asymptomatic. And this qPCR confirmed our transcriptome data, what we are seeing in the transcriptome data. And then we went ahead to look at the auxin biosynthesis, and we found some of the interesting genes which are involved in auxin and biosynthesis, like the TAR1 and UCAS, which are involved in biosynthesis. So as you can see here, we're expecting the UCAS, they are manipulated because some of the UCAS genes are upregulated, some are downregulated. And when we went again to look at the auxin responsive gene, we found one of them, one gene which is AUXIAA, which is a negative regulator of auxin, and it was upregulated in both conditions, that is symptomatic and asymptomatic. And then when we did the qPCR on auxin high AA, we found that the levels of auxin high AA between asymptomatic and symptomatic levels are the same. So this confirmed it matched with our transcriptome data. Then we went ahead to look at the inactivation. We found three genes which are involved in activating the active IAA or auxin to inactive form, and we found two genes which were upregulated, and these are Gretchen genes of GH3.6 and 3.9. They were upregulated in both conditions. And these genes are involved to convert, as I have said, auxin to amino auxin. And the takeaway from this slide is RLV might be manipulating the auxin and converting it to storage form. That's why we don't see, when we measure the level of auxin, we don't see it being upregulated in each condition because they are upregulated in both conditions. Then the other takeaway we can take from this slide is the commit gene or commit gene expression levels, maybe because it's high on symptomatic compared to asymptomatic, maybe the one which may be leading to stem thickening, which we are seeing in certain tissues compared to asymptomatic. So we went ahead to look at the crosstalk or the crosstalk between the 18 genes which are involved in housing and gibberellin levels. And we found there is a lot of crosstalk between these 18 genes with other hormones. And it was surprising to see the GA or gibberellin is the one which is controlling the cis element or the cis element in the promoter region of those genes. And the lowest cis element of GA was 4, and the maximum was 19. And then when as you can see as well again in the QPCR which I've shown before, the GA levels are more unasymptomatic, where we have the shoot compared to symptomatic. And then when you look at the cytokinin, the levels is the cis element, they are either 0 or just 1 to 3. So there is a crosstalk which is happening between these 18 elements and at the cis, the promoter region. So the takeaway from this slide is the GA is most likely to play a pivotal role in the setting, and this data suggests disease is getting influenced by manipulation of cytokinin, GA, stigoractin, abscisic acid, and housing by NLP. So when you look at the caffeic acid levels here, and the two genes which are involved in caffeic acid production, it is, as you can see here, GA level, then it's the cis element of the promoter region is 12, and 10, that is high. And then we look at the abscisic acid, acetokinin, and stigoractin biosythesis signaling, how they are manipulated in the affected plant. So when we manually look at the genes which are involved in the ABA, cytokinin, stigoractin, biosythesis and signaling, we identify 19 genes. And these 19 genes we found, or when you look at the biosythesis of cytokinin or zeatin, you can see some of these genes are downregulated. And then the surprising part, because we see them being downregulated in biosynthesis, we expecting them for signaling to be downregulated as well. But surprisingly, it is upregulated. Then we went ahead to look at the ABA, the genes which are associated with ABA biosynthesis and signaling. And you can see here the NCT gene which are involved in biosynthesis of ABA, they are downregulated. They are downregulated. But when you come to signaling, we expecting them to be downregulated, but the signaling is upregulated. And then there's the one of the most interesting genes is gas PC, which is involved in which is the negative regulator of APA biosynthesis, and you can see it is more upregulated on symptomatic. And then we went ahead to look at the stigoractin, which we found in our data that it is more upregulated in both conditions. And these genes matched with our geoterm, because they are these two genes which is involved in biosynthesis of the biosynthesis of stigoractin. They are upregulated in both conditions. So we went ahead to look at the GA biosynthesis, and the genes which are involved in GA biosynthesis, all those genes they are manipulated by NLP. We see some are either upregulated, some are downregulated, and NLP might be influencing these manipulation of these genes. So the takeaway from this slide is the virus manipulates the ABS, cytokinin, and stigoractin biosynthesis and signaling. As you can see, either biosynthesis is upregulated and signaling is downregulated. So we looked at again the promoter region, the cis element in the promoter region of ABA, cytokinin, and stigoractin genes. And what we found is there were more of the GA element which are controlling the promoter region of this gene. And then the second gene or hormone which is controlling the promoter region of this gene is the auxin gene, as you can see. And then the cytokinin levels, the cytokinin the cytokinin is controlling less compared to GA and AUXI. And these genes as well, as you know, they're involved in leaf development, flower development, and lateral flower development, where we see the rosette. So we went ahead to look at the transcription factors, which are associated with flowers, leaf, and lateral flower development. And we found 14 genes, 14 transcription factors in our data set. And these 14 transcription factors, they work as the apical or lateral meristem development, which lead to development of the new shoots. So we started looking at the as you can see in LOF1 and LAX1. This LOF1 and LAX1, they used to the LOF1 is used to help to establish and maintain the boundary cells. And then the LAX is used to control the initiation of the auxiliary meristem. And you can see here it is downregulated in both, is downregulated or manipulated in the infected tissue, that is in both symptomatic and asymptomatic. Then there was one most interesting gene or in transcription factor, which is CLAVATA1. So the CLAVATA1 was upregulated in symptomatic and downregulated in asymptomatic. And the work of the CLAVATA is to restrict the size of the meristematic cell population by negatively regulating the USHO, and the work of the USHO is proliferation. And the other work of the USHO is to maintain stem cell in the shoot apical meristem and promote stem cell identity. So when it is upregulated, the CLAVATA1 is upregulated in symptomatic, we expect the work of the USHOs to be downregulated because it enables the work of the USHO. So the other, as I have said, the other thing we look at is the USHO. So the USHO transcription factors, they are downregulated in asymptomatic, and then they are upregulated in symptomatic. And the USHO controls a lot the formation of axillary and lateral meristemic cells. So we went ahead, and then the takeaway before I go to the next slide is LRV manipulate the meristematic transcription factors, as you can see here, which may lead to abnormal leaf, flower, and lateral development. So we went ahead to look at the prickles and bones in symptomatic and asymptomatic shoots, and we found there is a morphological alteration of prickles and thorns in rosetted shoots. So when we went ahead, these are the pictures we collected the samples. So these are our pictures we took here. So the characteristic of the prickles which we identify in rosetted, they were large. As you can see here, they appear to be big in size. They are white, green, and brown coloration, and they are oriented perpendicularly to the stem, compared to the normal ones, which are not perpendicular to the stem. And when you have a close look, because you can see here, this is how it looks like. So they are very large compared to the asymptomatic branch. So when you look at the prickles in healthy and asymptomatic tissues, they were smaller, red, as you can see here, they're smaller, red or green coloration with oriented down at a downward angle. So the takeaway from this slide is the high virus data in certain tissues may be leading to manipulation of hormones and transcription factors involved in prickles and thorn development. So we went ahead and now we looked at the transcription factors which are involved in formation of the prickle and thorns, and we found they were manipulated because when you look here, the ROC3, which is involved in production of cytokinin, the ROC3 is downregulated in symptomatic. So we expect the levels of cytokinin or zeatin is low. And then when there is low levels of zeatin, we're expecting the MVW complex, which GRABRA1 is one of these complex, to be downregulated, and that is what is happening here. It is downregulated in both asymptomatic and symptomatic. But surprisingly, we're expecting, because when GRABRA1 is the one which activates the GRABRA2 and TT2, but when you look closely, the the GRBRA2 is upregulated in symptomatic and normal levels in asymptomatic. And we didn't know like which pathway is used in which other pathway, because the only pathway which is known to activate GRBRA2 is this pathway, but it was upregulated in symptomatic shoot, even though it is downregulated in GRBRA1. And then when we went to look at the transcriptome tester 2 or TT2, it was upregulated in both conditions, that is asymptomatic and symptomatic. But when you look at the GRBRA1, which is the precursor which activated, it was downregulated. And these are the genes, these are the how they upregulated the expression levels look like of these genes in the heat map. And yes, and then this is just a picture showing the pathway, or a figure showing how the upregulation of these transcription factors happens. So GRBRA1 activates GRBRA2, and then it causes cell differentiation for the formation of the dots. So the conclusion from this talk is LLV levels are high in lucetent shoot compared to asymptomatic shoots. And the levels of ABA, cytokinin, auxin, gibberellin, and stereoraptin are manipulated by LLV, as we have seen, leading to abnormal development. And then the LLV alters the meristemematic transcription factors, leading to deformation of lateral and apical bands. And the caffeic acid, acid is reprogrammed, as we have seen a certain shoot, leading to a production of lignin, which may be the cause of stem thickening. And then RLV, also we have seen it may be altering the transcription factors involved in frequent development, leading to formation of deformed precoz and dots. And then lastly, I want to acknowledge my advisor, Dr. Ashok, my Lami Shakil, and then our collaborators, Dr. Hong, Dr. Hoska, Dr. Venura, Dr. Holwitz, and Dr. Catherine, Dr. Van, and Jeff Freud. Can take questions.
Thank you. We've got a couple minutes for questions.
Yeah, given the level of expression of these genes in the symptomatic and the asymptomatic, so if trying to design a diagnostic method to stop, I would say to detect, if the material is susceptible or not susceptible, to which balance of these two genes you will look first?
Okay, so from this transcriptome, if I understand your question, so you're asking like if you collect samples from the field and you want to test if they are positive of RLV, which two genes can you focus on? Yeah. So I think on the virus side, we focus on the virus primer. That is the one concerned with RNA3, that is the one which we saw.
That's clear. I'm seeing the blank.
In the front.
Yeah.
So in the front, we have found genes which are yes. Yes, if you can see here, we have found these genes, caffeic acid is more upregulated on symptomatic and higher levels compared to asymptomatic. And when you look at the heat map here, the expression level of the same gene, that is the commit gene which is involved in production of caffeic acid, is more on symptomatic compared to asymptomatic. So we have confirmed this gene with the qPCR, and we have seen the expression levels is more on symptomatic compared to asymptomatic. So the other gene which we have checked is the SCRI domain gene, which is part of the auxin responsive gene, the greater gene factors, and we have found that the gene is more regulated in symptomatic compared to asymptomatic. And then when we look at this gene in our data set here, is as well upregulated in symptomatic compared to asymptomatic. So yes, those are the two genes which I can recommend.
But you don't like the transcription factor?
The transcription factors, Yes, the reason why I'm not talking about the transcription factors, we didn't do qPCR on them. So I'm talking about the one we have done qPCR and it has matched with our transcriptome.